So, good morning, everyone. Since on the last presenter, I hope we can end it on a good note. So, I'm Kiran Kumar. I'm from Biodiversity Research Center, Academia Sinica, and today I'm here to talk about my research topic, that is the evolution, pathogenicity, and the biosynthetic potential of the genus Nocardia. Now, genus Nocardia is a pathogenic bacteria, and it's known to cause disease, infections. But here today we are talking about the secondary metabolite it generates, and to find some new gene clusters which is responsible to generate these bioactive compounds. So we chose Nocardia because of its ubiquitous properties, because that means it's found in almost all the environmental conditions. It is yet an underexplored genus because of the complex taxonomy which it has. It is an opportunistic pathogen. It's known to cause disease, that is nocardiosis, which is very much prevalent in US, Taiwan, India. It is also known to produce more than 50 bioactive secondary metabolites, which have the antimicrobial, antibacterial, anti-cancer, and the anti-tumor properties. Now, such study can be very much useful for the medical and the environmental biotechnology. So here we see the methodology which we have followed. So we did the genome mining here. We downloaded all the genomes of the Nocardia from the NCBI database, and we collected more than 300 genomes. We filtered them by using taking only those species which have been taxonomically validated, and we have checked the genome assembly quality by using Busco scores. So overall, we have around 263 genomes of 88 species. And let's go one by one on the results. So the genome which were collected, that is 263 genomes, we wanted to know what quality of genomes we have, to analyze if we can have those genomes useful for the gene cluster which can produce the secondary metabolites. So basically, most of the genome which we have downloaded, they have been isolated from humans. Since it's pathogenic, so that means most of the researchers which are working on this Nocardia are mostly focusing on the pathogenic aspect of this, and mostly they assembled at the quantic level. Although they are assembled at the quantic level, they still have the high quality assemblies. We can see that from the Busco scores. The genome size ranges from 5 mbp to 10, so they have a very high variable in the genome size and gene content. In terms of gene content, they have from 4,000 genes per genome to 10,000 genes per genome. But the main point is that more than 50% of these genes, they're hypothetical proteins. That means their content has not been well defined yet. And most of the genomes, they have, when we checked about the valence content, they have this very much strain-specific valence content, and they are very much focused on the immune evasion, which helps the pathogen to survive in the human as a host. So overall, we can say that it has a distinct genetic makeup, and it is very much the genome size and gene content is highly variable. Now we wanted to know whether they have new genes or not. So we did the pan-genome analysis for all the 88 species. You can see, hold on, yeah. So you can see this, the core genes. We have around over 800 core genes, and we have found around 70% of the new genes. That means the more the number of genomes are sequenced for this Nocardia, the tendency of finding the new genes on a much higher scale. And it can also be proved by the Heaps law. So the alpha over here is less than 1. So basically, in future, the number of genomes which can be sequenced for the Nocardia, the tendency to find the new genes is much more higher. But the core genes are very much lower. We have around 800 core genes overall. So overall, we can say that since the core gene is low and the new genes are high, it's an open pangenome structure. And due to the finding of new genes, a much more higher scale, we can say that it has the genome plasticity is much more higher scale. Now, since we know that it has new genes, we wanted to know whether these new genes does have the potential to produce the secondary metabolite. So we identified those gene clusters which have biosynthetic potential. Those are called as BGCs. So we have found over 8,000 BGCs which are responsible for making the secondary metabolites from 46 different types of classes, from non-ribosome polypeptides to terpenes to ectoins. So here we have done the phylogenetic analysis for all the 88 species along with the average count of this for 46 different classes of BGCs. So our study will be the first one to report the two BGCs, that is lenthopeptide class 5 and proteosin. Now literature suggests that this proteosin has been known to have this anti-cancer and anti-tumor properties, but it has not been reported in the Nocardia species yet. So we'll be the first one to do so. Based on this tree, we can say that the gene cluster which are responsible, they have very much species specific diversity, and that reflects their gene turnover. We did the network analysis by you taking all the 8,000 genes. Here we can see that each dot is a BGC, and each line is a connection between the two BGCs. For example, if I show this terpene BGC, this is a gene cluster family. So this is entirely of the terpenes. Now here, if you zoom more closer, you will see this one is a BGC, and this one is the reference BGC. Now this reference BGC means that it has been experimentally validated and it has been proved. So now this shows that the Nocardia genome, which we have BGC from our data, has a potential to produce that kind of secondary metabolite which has been already experimentally proven. Now, some have gene cluster families clusters and some have this singletons. That means they can have both. They can have the shared pathways and they also can produce some novel biosynthetic capabilities that we are yet to study yet. But overall, among these classes, the Nocardia non-ribosome polypeptide and the hybrid of type 1 polypeptide synthase have been the most to have this reference BGC. That means they can produce the experimental validated compounds. One of this we have been shown for the Nocobactin. Nocobactin is known to have this anti-cancer anti-tumor properties, and this is the cluster of this. Most of them, they belong to this Nocardia falcinica genomes. We can see here more than 5 different species of Nocardia have been known to produce this this Nocobactin compound, which has not been reported yet. And that means all the other 5 species can also produce this Nocobactin compound other than Nocardia falcinica. And I'd like to thank all my collaborators. Thank you. I'm sorry.